Did you know that for decades we've believed our bloodstream to be a sterile environment, a closed system entirely free of bacteria? Well, recent research is starting to challenge that long held assumption. In our latest study, we set out to explore a bold question. Could there be a community of microorganisms, what we call a microbiota, living in the blood of patients with inflammatory bowel diseases like Crohn's disease and ulcerative colitis? To delve into this question, we examined blood samples from individuals managing their inflammatory bowel disease as outpatients. These patients live with their condition daily, but are not hospitalized. They come to the hospital for regular checkups rather than emergency treatment. Importantly, they weren't experiencing severe flare-ups or acute symptoms at the time of our study. By focusing on this group, we aim to discover whether a blood microbiota exists during the more stable phases of inflammatory bowel disease. We know that the gut microbiota plays a significant role in health and disease, especially in conditions like inflammatory bowel disease or IBD. But the idea that bacteria might circulate in our blood, that's a frontier that's only beginning to be explored. Using advanced sequencing technology, which allows us to identify bacteria based on their genetic material, we analyzed blood samples from these IBD outpatients. What we found was fascinating. Contrary to the traditional belief of a sterile bloodstream, we detected a diverse range of bacterial DNA in the blood samples. Notably, we observed a high proportion of Proteobacteria, a group of bacteria not commonly found in such abundance in the human gut or mouth. But here's where it gets even more interesting. We discovered that the diversity of the blood microbiota was positively associated with levels of C-reactive protein, or CRP, which is a marker of inflammation in the body. This means that patients with higher bacterial diversity in their blood also had higher levels of inflammation. So, could these bacteria be contributing to the inflammatory processes in IBD, or are they simply passengers hitching a ride due to the compromised gut barrier in these patients? One of the challenges in this kind of research is ensuring that we're not just detecting contaminants from the environment or the lab. To tackle this, we included negative controls throughout our experiments. While some bacteria were present in both our samples and the non-template controls used to detect any contamination, certain bacteria were found exclusively in the patient's blood samples. Unfortunately, we did not have samples from healthy individuals, so we couldn't determine whether these bacterial populations are unique to IBD patients or also present in healthy people. Including such healthy controls in future studies will be crucial to establish a baseline for comparison and to better understand the significance of these findings. So, given these findings and limitations, what does all this mean for patients and the medical community? Our results suggest that the blood microbiota might play a previously unrecognized role in the inflammatory processes associated with IBD. This opens up new avenues for research and potential therapeutic strategies. Could manipulating the blood microbiota help in managing inflammation? Could specific bacteria serve as biomarkers for disease activity or progression? Of course, there's still much we don't know. However, one thing is certain. The discovery of a diverse blood microbiota challenges our understanding of human biology and disease. It highlights the intricate connections between different systems in our bodies and the microorganisms that inhabit them. Thank you for joining us on this journey into the microscopic world within us. If you're interested in reading our original research paper, you can find the link in the description below.